Thank you.
Over to you.
Thank you, Sarah. And thank you all for inviting me here to talk this morning about why consider evolution in nutrition. Well, the answer was given by J.F. Kennedy, so we celebrate the past to awaken the future. Well, one of the most interesting things about biology is the focus that we have on the gene-centric paradigm. And this is a photograph 51 that Rosalind Franklin took, which was the actually giveaway about the structure of DNA. And this led to Crick's central dogma that you had DNA going to the cell. You never had it going back from the cell to the DNA. It was that was the central dogma, which has lasted for quite a long time. Now, the interesting thing about this is that this is much of it cemented by concepts of natural selection. But very few people seem to know that Darwin actually, in all 6 editions of his book, referred to 2 forces in evolution. One was natural selection and the other was conditions of existence. And of the 2, he said, the conditions of existence was the most powerful. Now, unfortunately, what happened was a fellow called August Weismann came along and said, this is far too Lamarckian. And so he cut the tails off rodents, and several hundred rodents. And as they sort of grew tails, in the next generation, and the generation after that, and the generation after that, he wrote this thing called the all-sufficiency of natural selection. And that dumped Darwin's conditions of existence. And from then on, you had neo-Darwinism, where evolutionary biologists thought only in terms of natural selection and tried to justify everything that happened in the context of the paradigm of natural selection. But Darwin actually said very clearly there are 2 forces in evolution: natural selection, conditions of existence, and the conditions of existence are the most powerful. And it's pretty obvious, actually, when you stop and think about it, that that is true. Anyway, so this was Crick's central dogma all over again. And that was, so I want to just give you one very good example of conditions of existence as they apply today. It snows, this is at Mount McKinley in Alaska, and it washes right all the way down to the estuary, which is where the marine food web takes off in earnest. And here's our friends enjoying the Frutal del Mare. Now, the problem with that, of course, is iodine deficiency, because the soil has been washed free of a lot of trace elements, particularly iodine. And iodine is the most significant way to produce mentally retarded children, iodine deficiency. And there's between 1.5 to 2 billion people, according to WHO, currently at risk to iodine deficiency. And this is a very, very good example of conditions of existence. It's incidentally, as all of the people who have iodine deficiency are inland people. You don't find it in the fishing villages in Indonesia or Kerala or anywhere like that. But there's an example of extreme iodine deficiency in one of my photograph taken by one of my students in Sudan. Now, iodine, of course, goes along with the marine food web. The richest food resource for iodine, like docosahexaenoic acid, is the marine food web. So let's look at the chemistry of this thing. Now, I hope you all by now know about the metabolism of the essential fatty acids. I'm showing this because I want to point out the slows and very slows. And the point about this is that very small mammals, like rats and rabbits, can make docosahexaenoic acid. Even so, it is a rate-limited reaction. The process is slow and rate-limited. And so, as growth velocity increased, it gradually outstripped ability to make docosahexaenoic acid. And when you look at the big mammals, as I'll show you later, you find that they can make very, very little docosahexaenoic acid. And with that, tumbles relative brain capacity. So this question of the slowness of the metabolic sequence of events, the rate limitations of it, which particularly applies to docosahexaenoic acid, more so than arachidonic acid, is absolutely critical to the thesis of how we actually got here. We came across this by studying, we wanted to know why animals had different sized brains. So we looked at the chemistry of the livers of some 42 different species and looked at the brains of the 42 different species. And the liver is all over the place. And you could actually tell how the pattern of the fatty acids in the different species was actually related both to growth velocity and to the diet of the animals. But when you looked at the brain, all that variability vanished. And that told us immediately that the brain was made to a very specific composition in terms of for its growth and function. And we did a lot of work to demonstrate that in the early 70s. But I'll not go. The one point I want to make again is when you see this process of rate limitation as applies to humans, when you look at how the fetus gets its docosahexaenoic acid, nature has abandoned all metabolic sequences and has gone for the specific selection of docosahexaenoic acid at each time it crosses a membrane. Each time it crosses a membrane, you get an enhancement of docosahexaenoic acid in the 2 position, the SN2 position of the phosphoglycerides. And so when you look at the maternal, cord, fetal, I mean, these are human data taken at mid-term elective abortions. You can see this biomagnification process that takes place, to which nature says, I'm not bothering about metabolic sequences, we just go for very specific selection every time we reincorporate docosahexaenoic acid. And this will come important again. Now, back to Darwin's conditions of existence. This is a kind of schema of what actually happened since the beginning of life. For 2.5 billion years, there was virtually no movement in the sense that it was all anaerobic prokaryotes. They excreted oxygen, of course, and eventually at some stage with photolysis of water and all that kind of thing, you got enough oxygen for air-breathing life to come into existence. And when that became thermodynamically possible, which was at the Vendian-Cambrian border, when that actually became thermodynamically possible, all 32 phyla that we have today come into the fossil record. All 32 phyla come into the fossil record straight away. And there's this brilliant Darwin's conditions of existence all over again. Without any question, this is the conditions of existence. And so here we have something really quite important, because if you look at the fossils of the pre-Cambrian era, the first 2.5 billion years, there's no sense of any kind of internal structure. There's no sense of internal structure for 2.5 billion years. There's just a globule. But you get internal structure when oxygen comes into the picture. And here's another condition of existence, because what is internal structure but membranes? These are lipids. The advent of oxygen made it possible to produce molecules which were really quite difficult to synthesize under anaerobic conditions. Take docosahexaenoic acid, for example. It has 6 double bonds. You need 6 oxygen atoms just to remove the hydrogens for those 6 double bonds, never mind the synthesis of the 22 carbons. So, unlikely that it was present before we really had significant amount of oxygen available. But the point really about that picture is that what effectively it shows is the significance of lipids in compartmentalization inside the cell, the mitochondria, even within the mitochondria. The organization of the electron transport system is organized by membranes. The reticuloendothelial system, the nuclear envelope. Say nothing about the plasma membrane and all that kind of thing, where all the stuff goes on. So it seems to me that something we need to think about is the probability that lipids, by organizing cells, actually contributed to the specialization within a cell. And why not then contribute to speciation? And it seems to me there's a question that needs to be answered about the role that lipids played in the Cambrian with the origin of the 32 phyla. But that's another talk. Now, back to docosahexaenoic acid. If you look at the dynoflagelate, it has an eye spot. It was sort of representative of things that might have lived about 600 million years ago. And eye spot contains not just docosahexaenoic acid in its phosphoglycerides, but di-docosahexaenoic acid. The same as in your eyeballs, same as in your photoreceptor. Now this is phenomenal because, in fact, what I suggest happened was that you had in the first 2.5 billion years, what you had was photons being converted into carbohydrates and proteins and things. Now what you have are photons being converted into electricity. And by converting photons into electricity, it caused movement and all sorts of things to happen. And converting photons into electricity sparked the evolution of the nervous system and ultimately the brain. And, well, there you go. Now, the interesting thing about the photoreceptor is I just want to draw your attention to the fact that the light actually comes in what a lot of people, including Richard Dawkins, say comes in at the wrong end. The photoreceptor actually has a head buried in the reticular endothelial system. And it's completely counterintuitive that light actually comes into the photoreceptor at the back end, not at the front of the photoreceptor. We'll come to that later. But there's the chemistry of the photoreceptor in blue, and it's greater than 50%. You can't get any more DHA into the photoreceptor phosphoglycerides. There was DHA in there as well. But the interesting thing is, if you look at the green or blue or whatever you like to call it, the sort of turquoise bar, that's what you find in cod muscle phosphoglycerides. Now, everybody talks about fish oils, how important they are, blah, blah, blah. Fish oils are fat. We dump fat in our adipose tissue. When you eat fish and seafood, you eat phosphoglycerides, and you eat stuff full of docosahexaenoic acid with very little of any of the other fatty acids. You can do the chemistry of nearly all marine fish and life, and it's very much the same. The phosphoglycerides is what you eat, so the use of fish oils, for example, is really a rather distant from what actually is the meaning of eating fish and seafood, which you see the meaning there. Now, okay, so what really you've got is that from the dinoflagellate, the cephalopod, the fish, the amphibia, reptiles, birds, mammals, and humans, the chemistry of the photoreceptor is identical. This is 600 million years of genetic evolution and change. But so far as the photoreceptor is concerned, and so far as the synapse on the neuron is concerned, the synapse neurons come later, but as far as they're concerned, chemistry is identical over 600 million years. Now, that to me is the most compelling evidence for the absolute essentiality of docosahexaenoic acid for the photoreceptor and the signaling systems of the brain. So there's just an experiment that we did with Ephraim Yovine showing that if you deprive a female of DHA or omega-3 fatty acids, these rats of course, during the development of the rat pups, that you can show that the provision of DHA allows neuronal migration in a good sort. Deprivation pretty well stops it, and not entirely, of course, but it reduces it. So what is so special about it? I want to take a sort of a bit of a trip down a different route. So what's special about DHA? Well, if you look at the molecule, there are two molecules, and this makes this 600 million year track record even more significant. If you look at the molecule, it has a sequence of double bonds that have got methylene groups in between each double bond. If you look at the docosapentaenoic acids, there are two docosapentaenoic acids differing only by 2 hydrogens. they miss out a double bond. Although one of them is a precursor, of course, for docosahexaenoga. So at the beginning, it would have been more available, but never used. All throughout these 600 million years, none of this stuff's used. The only one molecule that's been used is DHA. So this means to me that that 6 double bond sequence is absolutely vital so far as the signaling systems in which docosahexaenoic acid is involved in. And that's the alchemy optimized conformation. It's got 3 double bonds that are planar, which is quite interesting. It leads to certain quantum mechanical interesting effects. And here you see the electron density, the pi bond energy, is all alternating throughout the molecule, positive, negative, positive, negative, positive, negative, which is really very interesting because it raises a lot of questions. Now, the point about the double bond sequence being methylene interrupted is that if you look at the top level, the pi electrons are more or less confined to the wells of their individual double bonds. If you've got down at the bottom a conjugated sequence of double bonds, the pi electron clouds overlap. So if you put a positive charge and negative charge at one end of the molecule, the current goes zip like it was copper wire. But with docosahexaenoic acid, it's a resistor. Well, so how can a resistor be involved in signaling? I'll tell you. Now, this means you have to invoke the Heisenberg uncertainty principle, because what effectively you've got is that the electron can exist in both as a particle or as a wave. Now, as a particle, it can't go through these so-called energy barriers. But as a waveform, it can go anywhere. It can end up, Feynman says, you've got to consider all possibilities as to where an electron might end up. If it goes through an energy barrier, burrows, they call it, tunneling through the energy barrier, pops up the other side, and it finds the next double bond has got 2 pi electrons, both with opposite spins and of different quantum numbers, so it can't stay, it goes back again. But if you take 1 electron out of those double bonds, then an electron can move in, and provided it's got a different quantum number to the electron that's existing, can stay. And so you can actually get electrons to tunnel through the whole of that sequence. And this is like semiconductors, which you use to make your telephones work, and TVs and all the rest of it. So we've actually got here a potential for semiconduction. And the beauty about this is, and I'm coming to an important issue here, is about the theories of brain function, how the brain works. All the theories of brain function, including Roger Penrose and Hammerhoff, are all to do with protein tubules in case of Penrose and company. Protein has a different job in the brain, and that's to produce the huge amount of energy that it uses. Brain is made of fat. So what we're proposing actually is that the docosahexaenoic acid forms a sea of pi electrons in the synapses, the densely packed synapse, and then that is the key to the way the brain works. Because it provides this concept of using the Heisenberg principle and the polar exclusion principles means that you have absolute precision of the signal. And none of the guys who talk about proteins have got any idea about precision of signal. And you have to have precision of signal. Because if you just think for a moment, if you have 2 photoreceptors and the same wavelength hitting them both, but they respond differently, your vision is going to be garbled. And indeed, in fact, this is what happens, because if you induce an omega-3 deficiency and actually reduce the amount of DHA in the photoreceptor, the thing that's been universally discovered by people who do this sort of stuff, that you lose visual acuity. And that's the reason why you lose the visual acuity, because you're losing capability for precision. So precision is an absolutely sine qua non. You cannot have brain function, you cannot have a neural pathway operating without precision, otherwise it'll wander all over the place. You cannot have photoreception without precision. And indeed, the people who study this sort of thing have found that firing a photon, a single photon, at a photoreceptor will induce an energy output which is identical regardless of the wavelength that hits it. So precision is the key issue. And if you, Hopfield has calculated the distance for electron tunneling in this sort of way, and it falls off logarithmically with distance. And the problem with the precursors, the docosapentaenoic acid, is that whilst a 4.2 angstrom distance is fine for electron tunneling, 8.4 angstroms is just no go. So this explains why none of the two precursors or two docosapentaenoic acids were never used. Now let's look at why does the photoreceptor face the wrong way? Well, the reason for that is very simple. Because in fact, what happens is that incoming light hits a sea of backends, a forest of backends. And I don't know if you know about the Young's dual split experiment, which sparked off the beginnings of quantum mechanics. But what he did was to fire photons at two slits, and a photon should go through one or the other, but it doesn't. It in fact forms a wave interference pattern on the receiver. And so in effect what we're saying is that the system can operate as a waveform as well as a particle. And by converting it into a waveform, by the light coming in the back ends, what you have are wavelengths going through the photoreceptor. Now, just think about it. According to the classical system, the actual mechanism is that a photon hits an electron in retinol, in one double bond, the 11th cis position, activates the electron, takes it out of the orbit, and it falls back into the trans position, so you get all trans, and that's the signal that starts the whole ball game going according to the classical concept of photoreception. Now just think about this. A photon is traveling at the speed of light. A pi electron is whizzing round a couple of carbons at a phenomenal speed as well. Now how difficult is a particle going to be to actually find that single, not just any pi electron, but the one at the far end of retinol in that one double bond? How's it going to do it? But as a wave, the chances of it hitting something just depends very much on the wavelength, but all the visible wavelengths are going to ensure activation rather than as a particle. And that to me explains why the photoreceptor is the wrong way around. It turns everything into waves. So that now brings us into how actually does the brain work. Now the brain prize this year was given to Bliss, Coringery and Morris because they showed that memory was associated with the increase in protein in the synapses. There was very clear evidence about this. They did a lot of work on it. And the London taxi drivers, when they're learning the knowledge of London, they actually get enlarged bits of the hippocampus. So it's quite clear that learning process is associated with the growth or enhancement of these things. Now, the problem is that they pinned everything like Penrose and Hammerhoff on proteins. I've put to you that the job of proteins is to produce the phenomenal energy that the brain uses. You know, 20% of the energy consumed by you is being used for keep your brain going, although it's only 2% of your body weight. So, learning involves repetition and turnover. Docosahexaenoic acid is selectively incorporated during turnover. You saw that with the biomagnification process. So that each time it turns over, you get an enhancement of the docosahexaenoic acid in the synapse. Docosahexaenoic acid not only produces an increase in the density of pi electrons in the synapse membranes, but it also stimulates protein synthesis. This is well known by Conjugation company. And the other interesting fact that's actually quite little known is that Rappaport and company showed that the 26% of the energy used by the brain, that's more than a quarter of the energy used by the brain, is used for fatty acid turnover. So in fact, during the learning process, you know, for fair daffodils we weep to see thee haste away so soon, as yet, etc., etc., etc. To learn that, you have to repeat it time and time again until you get it work perfect. And learning, you see somebody in a room, and what's happening is photons are coming in with the image of the face. And David Marr has shown how the image actually is recorded in the brain itself as a kind of image that can be interrogated. So the plausibility is that docosahexaenoic acid, by being enhanced during turnover, stimulates protein synthesis, as shown by Bliss and company, and also enhances the pi electron density, enabling a pathway to be generated that once you get this chaotic flow of electrons coming, when they hit this pathway, they go down that route rather than somewhere else. And so you get recall of a memory and so on and so forth. And this is very much the same sort of thing, that showing the way that docosahexaenoic acid is actually selectively incorporated from the food to maternal plasma, fetal plasma, liver, fetal brain, the synapse, and finally the photoreceptor, which is the beauty about the photoreceptor is that it doesn't have time to learn. It's not a learning process. The photoreceptor has to work first light. No, that's why it is totally saturated, whereas the synapse is not. It has room to learn and grow its DHA and pi electron content. And of course you can forget by the whole thing degenerating. So that's the gene expression data. We don't need to go into that. Now the other thing is we all know about this. This is induction. This is induction. This is the way that dynamos work, that electric motors work. That passing a current in one direction can induce a current in another receiver. Now we know that. We also know from experience, for example, if you play the piano, you press C, middle C, and you not only hear middle C, but you hear a whole lot of vibrations, harmonics. You would press the soft pedal, so you only get C. Take off the soft pedal, press C again, and you get all these harmonic vibrations. If that works with sound, the same thing is going to work with these waveforms in the brain. I don't think there's any question about it. This is the way the brain is working. It's working by a function of waveforms directed through docosahexaenoic acid and other things. So there we go. That's just okay. So the recall process is another way in which the visual input from a face, for example, can create an electromagnetic enhancement of a model, a Marr-type model. But you hear the name coming in through the auditory system, and that will then, because it's simultaneous, have some kind of identity in terms of waveforms. And so when you see the face, it will harmonize with the name, and vice versa, and so on. So I think we can work it out that way. Now, there's a very interesting little additional point I'd like to make. That these studies on hallucinogens imply these are hallucinogenic type drugs. Aldous Huxley wrote this book, The Doors of Perception, the Gates of Heaven and Hell. And the interesting thing is that everybody considers that this is an enhancement of some kind of perception. Enhancement of these drugs are enhancing perception. And, but interestingly enough, psilocybin has been shown to studies to generate hyperconnectivity between neurons in places that you normally would not expect this to happen. And I just leave you with this thought, because if you look at serotonin and psilocin structures, they're pretty identical. So serotonin, as you know, has got all to do with a whole bunch of stuff of mood and cognition and so on and so forth. So if we look at the spin density, you can see that psilocin actually has 4, whereas serotonin only has 3 active regions where you get this strong positive-negative going on. And at the end of both molecules, you have a methylene interrupted between the aromatic stuff and the nitrogen at the end of the molecule. So in effect, what psilocin can be doing is actually acting as a better conductor than serotonin. And so what is happening is that the brain is being flooded by psilocin rather than just being kept calm and normal by serotonin. And it's the flooding that gives you this heightened perception that Aldous Huxley wrote about. So let's talk about human evolution, its dependence on the marine food web. Very briefly, how do we get from that little fellow to that girl? Well, I talked about how big animals lost out and were unable to make DHA. Here's a very good example of it. Weighs a ton at the age of 4. Human child, look at the hand of the child compared to the hand of the mother. The brain cases are almost similar. The priority in Homo sapiens is the brain. There's no question about this. Absolutely no question about it. It's not body growth. It's not body growth. It's brain growth. The Savannah hypothesis of our origin is that brain enlargement occurred from fierce competition with the top carnivores. It's led to the expansion of intellect, and you've got all sorts of theories about endurance running and so on and so forth. Upright stand will need to see over tall grass and throw spears, all that sort of stuff. Hairlessness from males choosing to copulate with females. Hair on the head to protect the sun. And Leslie Aiello said, the editor of Journal of Human Nutrition, no fossil evidence of a coastal origin. So any idea that DHA was involved in human evolution was more or less rubbished. Well, the aquatic ape hypothesis, or the waterside hypothesis, talks about, this is Alistair Hardy way back in the 1960s, the physiological similarities that we have between marine mammals is quite striking. Subcutaneous fat, diving reflex, hairlessness, sweat glands, and bipedalism in the sense that they have developed a supine shape. And we actually have got a supine shape as well. Now, the other thing about this hunting business that they talk about, you don't stand up to throw spears if you want to hunt an animal. You crawl to get close to it. And I mean, that's just simply ideas of people who have never been in. Now that's the chemistry of the brain. Predominantly docosahexaenoic acid, very little of the other omega-3s. Arachidonic acid from the land-based food web is important as well. That's what happens to these large mammals. You see, they run out of DHA. They can make the 22 carbon with 5 double bonds, but they can't get much further. So these have all universally got small brains. There's the sort of sequence of the brain size as body size increases, brain size collapses without exception in all land-based mammals. And that's all that's the brain that's inside the rhinoceros's head. Put the dolphin back and Homo sapiens into that picture and you get another story. We don't actually have a big brain. What happens is that we have retained our brain capacity that we had as a small mammal. Therefore, we had to have an ecological niche that provided docosahexaenoic acid, iodine, other trace elements that were essential, and that has to be the seaside, put it bluntly. Dolphins, well, there you are. If you compare the dolphin chemistry with land-based mammal, it's chalk and cheese. it obviously gets all the DHA it needs from things. And we now have fossil evidence quite clear from Curtis Marions and John Parkington. We have identified the earliest species of appearance of dietary, technological, and cultural package that included coastal occupation, platelet technology, pigment use, and dietary expansion to marine shellfish, and is dated to a time close to the biological emergence of modern humans. That's 160 to 180,000 years ago. That's Curtis Marion. And he doesn't know anything about DHA. Or he didn't. He's beginning to learn. That's a picture of where they were working. Coastline just stuffed full of food. And Herto is quite interesting because the brain size, the families found Herto is bigger than Henry's got today, which is, again, quite enough. Just to make sure that you understand this, because I think it's important, if I can find it. Okay. So here is Heidelbergensis. You know where Heidelberg is? It's far from the sea. This guy, Neanderthal, didn't eat fish. There's no frontal cortex. Okay, he's got a big brain, but he doesn't really have a frontal cortex. That's Heidelbergensis. Somewhere in here is Homo sapiens. There. Now, this is actually Cro-Magnon, but it could be Herto. But this is 160,000 years ago. This is about 60,000 years ago. Herto was 160, 180,000 years ago. Ate fish, no fish. Case closed. Okay, now, so there we are. That's the rock. You know, this is a cartoon of Homo sapiens as the savanna hunter and gatherer. There he is. Now, what I would suggest, this is a much more realistic image of painting of what people's imaginary was of Homo sapiens throwing spears at animals and things like that. I think this is a much nicer image of Homo sapiens during formation. So there you are. Chris Stringer shows that Homo sapiens populated the planet by wandering around the coastlines. And here you have Moken today. Even today, people still living almost in the seawater. They give birth to the children in the sea, and the children actually wean themselves by diving and swimming down to the floor of the sea to get food. That's how they wean themselves, diving and swimming. And there's quite a lot of these people. Now, this is interesting. David Attenborough had a program called the Scars of Evolution, radio program, in which all the people, the Savannah hypothesis people said, poured a ton of cold water on this as the aquatic relationship that we might have had. And so I and various friends supported the aquatic connection. And at the end of the program, he said, you know what, that's an interesting thought. This is typical of David Attenborough. He said, you know, I suddenly thought human babies are born with a layer of vernix, a waxy substance that covers the baby. He said, that sounds like, so something to do with protection against water and that kind of thing. I wonder if any marine mammals are born with it, because no land mammal is born with it. No primate is born with vernix. So 2 weeks before the program was due to come out, so he got a phone call from a professor in Vancouver who said, yes, the harbor seal is born with vernix. So I got in hold of Tom Brennan and said, look, Tom, he's an absolute super with mass spectrometry. Why don't you get some vernix from harbor seal and various animals and human vernix and see what they look like together? Well, they're bloody well identical. So, I mean, this is unbelievable. There's a close similarity with the vernix that covers the human baby and the vernix that covers 3 marine mammals that he studied at birth. Close identity. Well, I'm running out of time. Okay, now I'm not going to go into that. fish consumption, depression, the aspect study, direct line with the amount of fish and seafood and the intelligence of the children at 8 years of age. What have we done with our food? Well, we've intensively reared chickens, and look, that bottle contains the amount of fat out of one intensively reared chicken. Just that's fat that comes out of the thing. We've completely destroyed our food web. I mean, this is supposed to be meat. People say how good it is. And, you know, we saw this in 1972. The writing was on the wall even then. We wrote a book, and Graham Rose reviewed it in Sunday Times and said, if we don't do something about it, we're going to become a race of morons. That's in the Sunday Times. Over the past 2,000 years, volume of the human brain has decreased from 1,500 cc's, that's HER2. And our Dutch friends talking about intelligence quotient of the Western world has dropped by 14 points as compared to those living in Victorian times. I don't know how true all this is, but maybe Fritz will tell us about the thing. And, you know, and people are concerned about this. This is Einstein and Stephen Hawking. They're concerned about this stuff. We've got population growth. It is unbelievable. Population growth is now logarithmic. You know, 1804 to 1912, it took about 27 years just to add 1 billion. Now we're adding 1 billion in 10 years. It's unbelievable. Right, so this is the real crunch. The European Union did an audit of ill health in 2004, and brain disorders came top of the list at 386 billion. So everybody said, "Yahoo! This can't be right because it's you know, there's new diagnostics." Okay, do it again. 210. Oh, you've added another country. The UK did the numbers in 2000, 77 billion. In 2010, for the same reasons, Joe Nurse repeated it, 105 billion. And Wellcome Trust, 113 billion. And number 2, world burden. This is the problem. How are we going to feed this population with enough stuff to enable us to have children born into this world with enhanced neurodevelopment? It's going the other way. And conditions of existence will determine which way it goes. There's Beijing supermarkets, all full of European sausages and pork pies and God knows what else. All the excess high fat. This is lovely. Herculaneum, you know, was covered in ash, and they've been looking at the sewers in Herculaneum, and in Herculaneum they discovered that the Roman, even the poor people, were eating an incredible variety of fish and seafood. Now I want to show you the content of a sewer in London. A sewer in London was stuffed, jammed with fat, and it took them 2 weeks to remove it because people were alerted to this because the toilets weren't working. So, case stress. So in summary, the brain evolved in the sea using docosahexaenoic acid for its signaling. Homo sapiens could not have evolved on the savannas where all land-based mammals had little DHA and their brains shrank. In recent time, we've become increasingly dependent on intensively raised land-based foods, and that's the problem with the rise of mental ill health and brain disorders. It's the gravest threat facing humanity today. The future of our children and their children is at stake. We celebrate the past to awaken the future. Thank you very much for listening. No, we don't have time for questions. I hoped we didn't have time for questions.
So I could kick off and just say, in terms of government recommendations for fatty acid intake. What are your thoughts on those?
Well, my thoughts are that we don't want recommendations on fatty acid intake. What we want are recommendations on food. And the first thing that the government needs to do is to start to develop our coastlines, redevelop our coastlines as they once were. As we were talking earlier about how in London in 1900, the barman would go down to the Thames and fill the buckets with oysters and put them on the bar free for people to eat. There's been a complete, particularly after World War II. During World War II, fish and seafood was not rationed. Meat, sugar, all the rest of it was rationed, but fish and seafood was not. And there was no child during World War II that said, "Oh, I don't like fish." They wouldn't have had it to eat. Well, that's not true, but I mean, there simply wasn't any kind of, we have the radio doctor, we had a whole lot of stuff going on during the war, and fish and seafood was the only major food item that wasn't rationed. And we ate a lot of it. We even ate whales during the war. And after the war, they decided that this nutrition and health that Drummond had been on about during the war was not what we want. We want production, production, production. And that was the end of it, because the whole thing swung in favor of land-based food production. And so you now get a balanced change like this, and this is what's the cause of, in my opinion, the cause of the rise in mental ill health. It's very simple. And we've got to get back to cleaning our rivers so that they're no longer polluted, so that the estuaries, which were, as I showed you, once the beginning of the marine food web, can be restored to the health again. And you can get back to the oysters, mussels, crabs, lobsters, and the marine food web enhancing fish production. We've got a coastline. We've got no more arable land that we can exploit in this country. 70% of the UK is arable land, and they're not going to get another square metre practically. But we've got, what is it? Something like 19,000 kilometres of coastline.
I was wondering about, well, a lot of talk about the sustainability of fishing and all sorts, but then also, if you're willing to increase the coastline and the fishing production, I think farming fish is just as good in regards to the same nutrients that you can provide from free range or wild fish, yeah. So I guess there's two questions: the whole sustainability issue with fishing, and then also, can farm fish be the same quality?
I'm not quite sure what the question is.
So you're talking about more fish, not fishing, but more seafood production and
Restoring it.
Restoring it. But then also, isn't it a problem if there's increasing populations to have that?
Well, that's the reason for it, because I mean, the Foresight report, 2011 for government, there's 400 people across the planet said we've almost worldwide reached the limit of arable land development, and population explosion is going to erode that as well. And if that's the case, where are we going to feed people? How are we going to feed them? Only the wild fish catch reached a plateau 20 years ago. We're not getting any more fish out of the oceans. So population is going up. Fish has flattened. Aquaculture is going up, but it's now reached a stage where they're having to feed chickens because they can't get enough fish to feed the aquaculture on us. So that's going to destroy the aquaculture system. The only solution is a solution that people came up with 10,000 years ago. When they ran into trouble with this hunting and gathering stuff that they talked about. And what did they do? They developed agriculture. They domesticated plants and animals. Now, surely in this century we have enough intelligence to be able to start thinking about doing the same thing with the oceans. It's the only place we can get new food resource from. It's going to require new technology, new all sorts of things, but it's the only place it's going to work.
Yeah, two questions. One is, could you talk briefly on the conversion kind of plant-based, alpha-linolenic acid to DHA, EPA, and also the second question was about imagery from say from meat, say grass-fed beef quality.
Yeah. The conversion of alpha-linolenic acid to docosahexaenoic acid is possible in practically all animals that have been studied, with the exception of cats, the carnivores. But it is very slow. So very small mammals with high metabolic rates can make enough, and they have a brain-body weight ratio of about 2%. We have a brain-body weight ratio of about 2%. As you get bigger and faster velocities of body growth, so protein deposition as cells grow outstrip the speed at which the animals can make DHA. So the faster the velocity of body growth, the less DHA you can make, and the more you actually become reliant on an external source to maintain it. Now, the land-based mammals can't do it, so all brain's capacity just plummeted as body size increased. And Homo sapiens, the argument is, overcame this problem by living beside the sea and had the best of both worlds, the marine and the land-based food systems. So I think that's as far as the people have done all the isotope studies, both in animals and in humans, to demonstrate the poverty of our ability. Some people can do it better than others, and certainly vegetarians are able to do it better than non-vegetarians. But the bottom line is that it's a slow process and difficult, and you're far better off getting the preformed stuff.